AFAR has actually had a long-standing history in supporting young investigators and mid-career investigators in the biology of aging and age-related research. Going over 28 years has actually supported funding of over $124 million worth of funds going out to over 2,600 different young and mid-career investigators. We had a dramatic impact on a number of investigators, including several who are actually in this room. And I actually have to thank AFAR for my first grant as well as several others that had actually propelled my career in academia as well as beyond. We're holding a conference on aging here to actually focus on the heart. This forum has been used to actually focus on different aspects of aging in terms of cancer, the brain, endocrine manifestations, but I really thought it was important to get to the heart of the matter. Vast majority of individuals in the Western world will still succumb to cardiovascular disease, and with the ever increasing impact of the aging population in cardiovascular disease, it's a good opportunity for us to actually start to focus on the specific needs, the specific biology, the specific translational aspects that need to be focused on in the aging heart. We have a number of incredibly impactful speakers today who will be speaking about the biology of the aging heart as well as translational aspects there that will be impacting future and present research. Most of our speakers after our keynote speaker, Ed Licata, will be speaking for 25 minutes, and we'll have some time for questions and answers.
Thank you very much, Jay. Good morning, ladies and gentlemen. I hope no one is disappointed that I changed the focus of the talk a little bit, but it eventually does get around to the heart. So this is the menu for my presentation, and as you can see, it spans a whole lot of different ideas, from demography to epidemiology to philosophy to cardiovascular clinical issues to cell and molecular issues, and if there's time, to ask whether we could do anything about it. So I'm going to focus on arterial changes that occur with age and their implications in disease, because most of the cardiovascular diseases that we have in older persons originate in the vasculature. I didn't know that for the first 20 years of my scientific life. You've all seen this type of graph before, that life expectancy as a function of the decade. Up until about this time, the gains in life expectancy were increasing lifespan over the earlier years, but past this time we've been adding numbers of older persons to our society. So just how many people are there of that type around the world? The pink here shows countries where the population has more than 20% over 60 years of age. That's why we call it the old country. Marco, it's okay. But looking to the future, it gets even more interesting. So imagine the world in 2050, and those of us who are alive, well, maybe those of you who are younger will be alive then, and imagine. It's a pink world. Now, cardiovascular disease is a major problem in older persons. So we have an increased number of persons who are older, and we have the major diseases that accompany old age. Coronary heart disease, this is the Framingham data. This is the incidence as a function of age in men and women. All these graphs get sort of boring after a while, graphs of this type, because they all show the same thing, more or less. This is Hypertension defined as 140/90 in men and women over the age of 60, non-Hispanic white, non-Hispanic black, and Mexican American. You see between 60 and 85% of persons over the age of 60 have hypertension. That's an epidemic. We need a flu vaccine for that, I guess. And the type of hypertension that occurs is predominantly systolic hypertension, the circles that you see here. Other types of hypertension begin to fade away with increasing age, but this type becomes the dominant one, and many of my remarks will be related to that when we get into the looking at the old arteries under the microscope later. So stroke increases exponentially with age in men and women. It ain't good yet. Hit it. Oh, go back. This is worthwhile making right. Can you hit it there and make it work? You did it before. Anyway, it works. Too bad. Okay, let's not worry about it. That was a plant to try to loosen you up. This was a hard audience so far. Anyway, this is a chronic heart failure as a function of age, and you see 14% of the population having a diagnosis of heart failure. That results from those diseases that I just mentioned, coronary artery atherosclerosis and high blood pressure. But clinical medicine hasn't really paid a lot of attention to age. It's been essentially ignored because many believe it's a non-modifiable risk factor, even though it's the major risk factor in any study. If the true population is sampled, it's thought not to be modifiable or just due to an increased time of exposure to other risk factors. And this increased time has become very important to me as I dwell on it recently. The world is not so simple as people made it out for me or painted the picture that I bought earlier on. I'll try to show you that. So what's aging? Okay, so let's do a reality check. So as I thought about it more and more, this is all there is to reality. It's hard to believe. You can't imagine that that's all there is. But if we consider what's the environment, then I think it'll become easier to understand. So we get DNA. It has an environment. The nucleus has an environment. The tissue is an environment for the cells. We have organs that communicate with each other. We have the organism. And I put personality in here because it's so important. And sometimes it gets shoved out in right field, but right here it's right in the center of things. Personality, lifestyle, etc. We have the society, like you have to wear a tie to get into this joint. It was taken away from me. They said I didn't earn it. We have the earth. We have the surrounding atmosphere. So these are the types of environments, and they all interact with each other. And I don't think anyone ever thought about how much signaling has to go on just to keep us in a steady state like we are now. I think, well, let me not get ahead of myself. So genetic and environmental pressures work together to maintain steady state systems of these phenotypes that we'll talk about, and the general cell phenotypes. Some of the characteristics, prominent characteristics of them are listed here, and they're all maintained by this system of mutual enslavement, as I called it, of DNA and its environment. So again, what's aging? So this system of mutually enslaved DNA and its environment changes with time. This results in phenotypic changes in cells, tissues, organs, and the organism. So you can watch it down here now. It's getting a little dimmer, you see. Depending upon what you believe. So some examples of the time-dependent failing interactions within this system. Signals are changing, the ability to sense signals, the ability to transmit signals, and the reaction to signals. These are just general types of things I thought about of how the system could fail. There are other erudite papers on all of these issues, which are a little bit beyond my realm. So the resultant stages of the phenotype change with age, and sometimes we don't think about this as a continuous happening with time as aging. We call something up here something different, but it's kind of all going in that direction. Cells, tissues, organs, and organisms. Now there are many examples that have been found through a lot of different types of research with respect to changes that occur in molecules, in cells, and in tissues that include mutations in DNA, oxidative damage, accumulation of post-translationally modified proteins, cells dying, cells becoming proliferative when they ordinarily shouldn't be, cells getting old, etc. And I'm not going to go into all of the different types. You'll hear a lot about many of these specific mechanisms in some of the other presentations today. And there are some conserved phenotypes with aging across species. Humans, okay? And many of you will recognize this infamous list. And the mouse conserves, I guess the human conserved the mouse pretty faithfully. And as we get down to lower species, some of these phenotypes are preserved even all the way through this hierarchy. So those are the clues, I think, research of this sort pointing to these phenotypes, if we could understand them more fully, being important to understand aging. So the golden years are here. Okay, so in other words, aging is a shift in an organism's reality. Now, there's been a lot of controversy going back to the Greek philosophers, and this has been summarized very nicely by Hermann Blumenthal in 2003 in the Journal of Gerontology. And Nathan Schock taught me to think this way, that aging and disease are not synonymous. They're different processes, they're related, they have important relationships, but not inevitable and not the same. So then I would plan my research along these lines. Right, Gene? We would try to separate, and we'll get back to this later because it's one way to proceed, otherwise you're stuck. We try to separate aging from lifestyle, from disease, and we're realizing that each of these has a genetic component and that environment's important. On the other hand, Grimley Evans, that's Sir John Grimley Evans, puts it this way. It's hard to separate the undefined from the undefinable. In that case, we have a scenario like this. And the key word is interactions. Keyword, it's not either or, it's interactions. But it's daunting to try to tap into those. So I sort of got a bright idea to try to articulate it this way. There are time-dependent changes in cardiovascular DNA environment system. So those changes that are occurring with age in that system of DNA environment interactions that I talked about, they alter the cardiovascular substrate on which diseases become superimposed. So we get a different picture of disease with respect to severity and prognosis and prevalence. So from here on out, we can say aging or disease can be construed as shifts over time in the balance of the genetic and environmental pressures that cause the DNA environment system to fail. This is a cartoon, more or less, of that statement. These people believe that aging is a disease because they contrast it with health. This is what happens to endothelial cells with aging. In health, there's a balance between these types of damage and the repairs. And with aging, this balance is tipped due to some of those changes that were on that large graph that I didn't articulate in detail. So why does aging occur? If you think I can answer that, you're crazy. So we need to answer that, we have to approach the scenario in a different way than we have so far. So to understand why aging occurs or how age-disease interactions occur, we need to define those interactions at each level. That's daunting, and the mechanisms that underlie those. And then we have to integrate the information. And that's why it's so hard to define a cause of death in old age. It's because all of those interactions, the sum total of them fail, and you can't do an autopsy and find the answer. But instead of integrating, what do? We fragment. We fragment organisms into systems, like our meeting today on the heart. Of course, we have to fragment to a certain extent, or there'd be no progress. Systems into cells, losing sight of organs. Cells into departments of biochemistry, physiology, sociology, etc. Tom Kirkwood, the credit line happened to be missing from here, but puts it this way. We need a systems biology approach to ensure that we have a healthy old age. So what's systems biology? This systems biology could be defined in a whole lot of different ways. Some of them are very highfalutin, some of them are more understandable. I've given you two more understandable ones. Again, Kirkwood, systems biology could be described as an integrative approach, being something alternative to a reductionist approach that has dominated the research agenda for years and that gets all the grant money right now. So this could be this chart going from theories of aging, evolutionary biologists, to geriatric medicine. If we separate it at each level, then it's kind of a reductionist biogerontology. But if we could capture vertically facets of each one of these components into one theme for our research, we might learn something new. So another similar but different definition of systems biology means to bring together very different people and bring them close together into working partnerships. So this is my laboratory at the intramural research program of the National Institute on Aging. These are our experts. This is our system of research with respect to aging and age-associated issues of the cardiovascular system. So we start out by trying to identify salient features in humans, and then we have studies that relate to their hearts, to their vasculature, and to the crosstalk between the two. And then we try to, after we define salient features, use other models to try to work out mechanisms that we can't easily achieve in humans. Once we find out something about mechanisms, then we have putative targets for some type of translational research with respect to interventions. And you'll hear about some of these things later today in more detail. Not what's going on in my lab, but more cutting edge. Now, so we began then by trying to understand what do we know about arterial aging without a textbook diagnosis. And this is the standard approach I told you that we use in the Baltimore Longitudinal Study on Aging. This study just had its 50th anniversary last year. I think I've been there for 40. And these changes we found in the Baltimore Longitudinal Study on Aging, and they've been found in many other populations as well. There's a diffuse, now we're talking about, I want to relate this to what happens in larger arteries. And so we're talking about properties of central arteries. There's a diffuse intimal medial thickness, and in humans it's mostly an intimal thickness. And there's matrix there and extra matrix in the arterial wall, and the collagen has increased non-enzymatic crosslinks. The elastin becomes frayed. There's endothelial dysfunction. These tubes become stiffer, and that's what causes systolic pressure and pulse pressure to increase. This shows the intimal medial thickness in the Baltimore Longitudinal Study on Aging, and this thickness is not considering our atherosclerotic plaques. These people are highly screened to exclude cardiovascular disease, and if there is a common carotid artery plaque here, they're not included. But you see that this increases with age, and you'll see the same thing in animal models that don't get atherosclerosis. And the other point is not just the increase with age. Look at the heterogeneity among older persons. This is something we didn't learn to appreciate until after the fact. We were always concerned with comparing young and old. So there are persons who age very successfully with respect to their arterial parameters like this one. There's the usual, that's the line, that should be a curvilinear line, by the way, and there are those people who age unsuccessfully, and that's where the risk is. Arterial stiffness can be monitored very easily, non-invasively, by placing a probe on the carotid artery, on the femoral artery. This would be the stiffness of the aorta, measuring the time it takes for the impulse to travel from that probe to that probe, and measuring the distance and calculating the pulse wave velocity. And pulse wave velocity increases with age. This is aortic pulse wave velocity, in a very similar way, pretty flat until about age 50 or so, and then showing an exponential increase with this heterogeneity. And pulse pressure then, similar scenario. So we wanted to know, could arterial stiffness predict future blood pressure changes? And this is in the Baltimore Longitudinal Study on Aging. We measured pulse wave velocity at the beginning of the study and then tracked blood pressure over 10 years. And those in the highest quartile had blood pressures as a function of age as on the dashed line, and those in the lowest quartile on the solid line. And we also could show that pulse wave velocity predicted incident hypertension. So those that got future hypertension had pulse wave velocity along the dashed line, the red, and those who had persistent normotension in blue. This is the event-free survival. This is the highest, greater than 5.9 and less than 5.9. And pulse wave velocity was an independent predictor, different from systolic pressure, and even a better predictor than systolic pressure at the beginning of the study. We started to embrace genetics in a population study in Sardinia. And this is a founder population in which 6,000 men and women have been recruited from 3 villages in the province of Lanusei. And we've measured 100 phenotypes in that population. And there are some cardiovascular phenotypes. And we find a great deal of heritability in blood pressure, but in pulse wave velocity, intimal medial thickness as well. And at the moment, we are engaged in genome-wide association studies to try to find single nucleotide polymorphisms that could account for this heritability. So far, we expected to find SNPs that would account for more variance, more percentage of the variance than we have so far. There are some SNPs that have been found, like this one, in the collagen type 4A gene. This is important to arterial aging. You'll see why when we look under the microscope in a bit, and especially in women who have the CC allele pattern. But again, the extent of the variance accounted for by these types of single nucleotide polymorphisms is quite small, and other SNPs have to be found, or gene-gene interactions and gene-environment interactions, and it's a long, hard road for genetics to work out the total genetic component of any of these issues. Okay, so those people who age unsuccessfully with respect to the arteries, are they at risk, or more at risk for future cardiovascular events? And yes, they are, and epidemiology studies have shown that these aspects of arterial aging are independent risk factors. That means they're associated with cardiovascular morbidity and mortality, even when the impact of other risk factors is taken into account. And this is, I'll show you just 2 examples. This is arterial stiffness as a function of pulse wave velocity. This shows the 10-year risk for the endpoint, and the women and men, and these are stratified by their blood pressure to get rid of the blood pressure issue. And you see that the risk increases with pulse wave velocity at any given blood pressure. And similarly, here pulse wave velocity has been broken up into tertiles, and this is the future cardiovascular disease event. Highest tertile, lowest tertile. So the idea is that central arterial stiffness, we didn't talk about reflected pulse waves today. It's a very interesting phenomenon. These raise the central and peripheral pulse pressure. And again, we didn't discuss how the central arterial, how the pulse pressure could be transmitted to certain arterial beds that are unprotected. They can't protect themselves against pulse pressure, in the kidney and the brain in particular. And that's one of the reasons why central arterial stiffening and isolated systolic hypertension is so important with respect to risk. So, if arterial aging is a risk factor for arterial disease, then the factors that underlie the aging per se might be targets for treatment. What are some of those factors? So we're going here. Doing better than I thought. Our approach has been not only to study animal models, but when we could get the tissue, to study non-human primates and humans as well. And this is a formidable list of things, changes that occur in various mechanisms or protein levels. And the point that we're going to go through some of those in more detail, but the point of this slide is to match up the pluses and minuses. And you see these changes that we portray here in humans, what I've told you about so far, the diffuse intimal thickening, endothelial dysfunction, increased stiffness, luminal dilatation, occurs in all species, even in those who don't get hypertension or atherosclerosis. And this pattern that I'll show you about is a pro-inflammatory profile. And this is going to become very important with respect to aligning this type of research on aging with the research that other people are doing in other cell types. This is the diffuse intimal medial thickening of the aorta in a representative sample from an older person, about 70 years of age, and a younger person. See, this is the thinner diffuse intima. So the intima has cells that migrate there, and they divide there, and they make matrix there. They do a lot of nasty stuff. And two prominent characteristics of vascular smooth muscle cells from older, in this case rats, are an increased ability to migrate or to invade basement membranes. This is cell migration in a Boyden type chamber assay. You put a chemoattractant in the bottom chamber. In the top chamber you put the cells. You put a filter in between the two chambers. You could line the filter with an artificial matrix like Matrigel, and the cells have to chew their way through the holes, and you count the cells on the bottom of the filter. So you see, young cells learn how to do this with increasing cell passage. This is kind of like aging in vitro, if you will. But old cells, while they still retain properties they had in the aorta, in this case at early passages, are raring to go and have achieved their full migration or invasion capacity right from the word go. Similarly, old vascular smooth muscle cells proliferate to a greater extent than younger ones do. And this might be some sort of a paradox in your mind. When if cells are becoming senescent, we think of senescence in vitro as stop dividing. We also think of old cells becoming cancer-like. Okay, so we have age-associated remodeling of the large arteries that I beat you over the head with already. This is it. We talked about vascular smooth muscle cell migration and invasion, and now we have to deal with these issues of basement membrane protein degradation, etc. And one of the reasons why these cells can have this behavior is because they express in the arterial wall, in vivo, and in early passage in vitro, active matrometallics protease 2. And the factors that activate matrix metalloprotease 2 are increased with age, and the factors that prevent its activation are reduced. This is a membrane-bound activator, MT1-MMP. Plasmin is an activator. You'll see something happen here in a minute. Active MMP2 does not only, so MMP2 is an elastase, and it degrades elastin, but type 4 collagen is one of its main substrates, and type 4 collagen is in the basement membrane around smooth muscle cells and in basement membranes throughout the wall. So it's good at breaking down type 4 collagen, but it also activates other growth factors. So TGF-beta is markedly increased within the arterial wall with aging, the precursor form as well as the active form, and MMP2 is crucial for the activation of the inactive TGF-beta, and that works through TGF-beta receptor 2 through SMAD signaling to increase fibronectin and collagen expression. The messenger RNA for TGF-beta is increased as well. Calpain, another protease, is absolutely required for MMP activation and for MMP production, actually. MCP1, monocyte chemoattractive protein, a nasty player in atherosclerosis, is highly expressed with aging in the arterial wall, and it feeds into this system, as you see, for these blue arrows relating to migration and invasion. It would have been so much easier to do this with a proteomics array, but it took us 10 or 15 years to do these one at a time. Okay, so what you see now is that all of these things, and we put the PDGF beta here, are increased with aging, and they are downstream of angiotensin II AT1 receptor. Angiotensinogen is increased in all of those species we talked about. Angiotensin II, well, angiotensin I we haven't measured. Converting enzyme is increased, angiotensin II levels are increased, and ETS1, these are transcription factors. This is a nuclear transcription factor measured in the nucleus, increased with age, like ETS1. We'll get to this in a minute. There are other parts to this pathway that were not shown on the prior one, in that reactive oxygen species are generated by this type of signaling. NADPH oxidase produces superoxide that steals nitric oxide to make peroxynitrite, and it reduces the bioavailability of nitric oxide. If we infuse over the course of a month with a mini pump angiotensin II into young rats, this is the control, this is after angiotensin II, their arteries look like those of the untreated old rat. And many of those biochemical changes and cellular changes that we talked about are increased as well. So the whole point of showing you that was to remind you or inform you that the arterial wall in aging and disease exhibits this type of inflammatory pattern. And sometimes it's not called an inflammatory pattern, but it is. And there, except for a modest increase in white blood cells in human aorta, there are no white blood cells to be found. The endothelial cells and the vascular smooth muscle cells do all of this to themselves. Now, what if, this gets back to the aging versus disease issue, what if similar age-associated changes and pathophysiologic changes that relate to disease occur? Are they one and the same? Yes, they are. There they are. So this is the list, some of the components of which I've described in humans, monkeys, and rats. I've added rabbits here. Experimental hypertension and atherosclerosis in younger animals. The green means concordance, an increase with age. The red is concordance, decreased with age, and inconsistent results are in yellow. The question mark means we couldn't find any data. So to beg the issue of aging versus disease, let's just say that aging arteries are fertile soil in which the seeds of arterial disease flourish. And this puts it into a little more perspective. This is from Matthews et al. In atherosclerosis, people in atherosclerosis research now are paying attention to vascular smooth muscle cell senescence. And of course, they realize it could occur through time-dependent mechanisms with respect to telomeres, for example, or this is stress-induced premature senescence. So reactive oxygen species can affect DNA and mitochondria, and in response to all of these types of signals, the cells, these would be proliferating cells, as these are in atherosclerosis, senesce. Now, this pro-inflammatory phenotype resembles a SASP, that's senescence-associated secretory phenotype, that's been described in other cell types by Judy Campisi and her colleagues. And their model is DNA damage. So DNA damage turns on that profile that you saw, and there's more than one outcome of that profile. Senescence is one, but tumor promotion is another. And there's more to be learned about the special context in which this occurs compared to when that occurs. Again, this is an example of DNA environment interactions. And what I've done is to, I did a little homework. Actually, I had one of my best fellows do a little homework. This is the SASP. This should be SASP, described by Judy Campisi in human diploid fibroblasts. This is another recent publication in which vascular smooth muscle cells were aged in vitro, and then a differential proteomic scheme was made. And you see what they pulled out. And this is the kind of stuff that we've been studying in the arterial wall in vivo or early passaged smooth muscle cells in vitro. Very exciting concordance. And it's important also, as Judy Campisi emphasizes, that damaged cells in tissues have effects that go beyond just their own damage. They influence other cells by that secretory profile that I showed you on the last couple slides. Now, I wanted to end up by this part by telling you about a new molecule that may explain part of the riddle of why cells, this may be a protection against senescence, if you will. But it may be helping cells maintain their proliferative ability and maybe helping them to go on to become oncogenic later. This is MFGE8, that's milk fat globule E8. Sometimes it was called SED1 in the past. It has two EGF motifs. It uses a receptor with alpha V beta 3 or beta 5, and it has motifs that relate to clotting factor 8 and epidermal growth factor. That's the E8. And this is secreted with milk fat. It's a membrane around milk fat globules. And in the breast, it was required to negotiate the clearance of apoptotic breast cells as the breast would atrophy. But there have been many other functions for it, and we found some in vascular smooth muscle cells recently. And first, we found this in a proteomic search, and we found that it was highly overexpressed with aging in all of those species that we deal with. This is the human data. This is aorta. And we found that MFGE8 has these biofunctions. Remember I told you vascular smooth muscle cells, older ones, like to invade, they like to proliferate. And when we knock down MFGE8, you'll see they become to look senescent, more senescent. And this is the Boyden chamber that I told you about earlier. The chemoattractant is PDGF. Old cells invade that basement membrane better than young cells do. And when MFGE8 is silenced, when the RNA is silenced, this accessibility is abolished of the old to invade. And instead of showing you a whole lot of data, we also found, by the way, that angiotensin II turns on MFGE8. And MFGE8 then increases this pathway. It activates ERGs, and it increases CDK4, not shown here, and it makes cells proliferative, even cells that have slowed down their proliferation after many long times in culture, and it blocks this pathway. And I'm not going to show you the data, but if we knock down MFGE8, we see an increase in P21, P53, senescence type of beta-gal, etc. Can we do anything about what we've talked about? That was a little bit out of place. So this sort of, for our purposes today, sums up a message I wanted to leave you with. And lifestyle genetics and time at risk push aging arteries over the edge. So I like to think about it in this way. Aging is the major risk factor for cardiovascular diseases in our society and most developed societies throughout the world. These are the diseases: brain failure, heart failure, hypertension, atherosclerosis, etc. I like to draw this line and call it the clinical practice threshold. What's above the line we've called disease in the past. You can collect money for treating disease. What's below the line is kind of what I've been talking about today. There are equivalent matching stories in the left ventricle. Some people have called that normal aging or normative aging. I wouldn't buy that. Think about age increasing this way. I would call this the prevention stage for this, especially those who have accelerated arterial aging, and they can be found non-invasively with those techniques that we used to show you those data slides earlier. It's not part of clinical practice yet. It's at the stage of epidemiologic verification of, there's a dozen studies almost now that show those risk factors that change with age are independent predictors for atherosclerosis or hypertension or both. So the answer to that question before, can we do anything about it? The answer is yes. Fortunately, lifestyle is very important. Just brisk walking that's insufficient to change cardiorespiratory fitness can lower arterial stiffness by 25% in older persons, less than 85 years of age. There are other things besides exercise that you see listed here. This is a summary of a lot of the exercise data, and Doug Seals at Boulder, Colorado has done a lot of this work, not the exclusive investigator in this area, but this is from his review article. And aerobic exercise in particular can reduce the large artery stiffness that we talked about, has effects on endothelial function, some effects on changes in the arterial wall structure, and in many cases you don't see those effects from resistance type exercises. Chronic converting enzyme inhibition retards arterial aging. This was so many years ago. We're in the midst of similar studies now to measure those more specific molecular changes that I told you about. But this was a lifelong, beginning 6 months of age, treatment of rats with an ACE inhibitor. And you see how it delayed, in this case, the intimal thickening in the rodent model. Of course, that waned with more advanced age. And finally, I wanted to mention non-enzymatic cross-linked breakers. This is a cross-linked protein. This drug, which is a phenylthiazolium derivative, ALT711, there's a new name now, aligebrium, I think, acts like an enzyme. It breaks the cross-link and then can return to find another cross-link. And it's been shown to reduce left ventricular stiffness in the canine model, to reduce arterial stiffness in diabetic rodents, and this was our study in non-human primates. And then more recently, it's been shown to increase compliance. So compliance is the opposite of stiffness for our purpose today. In humans, these were people who had predominantly systolic hypertension. This is ALT-711. This is placebo. It was a 56-day study, baseline, day 28, day 56. These are two different ways to measure compliance, and they were both significantly increased. That means stiffness was reduced, and this effect didn't occur in the placebo. So I think it's time to treat accelerated arterial aging. Of course, we need more proof of principle studies before we do that, but I think it's time to do the studies. And so I think that future medical practice will have to prevent the age-associated epidemic and those diseases that we talked about today by finding out who's aging their arteries unsuccessfully. In other words, to prevent or ameliorate arterial aging, to prevent or ameliorate clinical arterial disease. Thanks for your attention.
Very nice. Ed, could you think of any exceptions to what I seem to be seeing in all the parameters you showed as a function of age, namely the increased variance is a function of age? Which suggests it was consistent with a lot of maybe stochastic effects. I mean, you mentioned nature and nurture, but not chance variables and all the signal transduction things. You can think a lot of room for noise or stochastic variation. But are there any exceptions to that? Increased variance with age in arterial pathophysiology?
There are, and I don't remember all of the parameters, but probably 90% of the parameters do what we saw here, and 10% have the same variance in young and old. I've never seen smaller variance in old compared to young. They have the stochastic theory, and cumulative damage and is a way to explain the heterogeneity. But at least now we have a chance to, with respect to what's happening at the arterial wall, you see this is different than measuring stuff in the plasma. So it's a whole new ball game. And in the face of this evidence about how dangerous these types of unsuccessful events, whether they're due to stochastic events in all of these molecules that we talked about. On the other hand, you see that at least many of them can be linked into a specific pathway. Then the issue is, why does that pathway get turned on to larger or smaller extents in different individuals? So I think we have to move from theory of evolutionary biologist into prevention with epidemiology and clinical medicine. Otherwise, we'll just be talking about the same thing over and over again, trying to figure out which theory of aging is the best. Without being explicit, I've tried to sneak in a theory that maybe takes a little bit from all of them, and you could still ask in the end, why do those time-dependent changes occur? And that's when I said I didn't know the answer. I guess I would be in Stockholm, but we could have listed all the theories then there at that point and said it could be this, it could be that.
Decline of the force of natural selection.
Well, sure, that's antagonistic pleiotropy. In fact, I had those slides in, but I took them out. I thought it would be boring for you.
Ed, while you're waiting for the proof of principle work to be done, one could make a public health epidemiologic argument that interventions to promote exercise across the population are really the solution. And if you want to look for a piece of suggestive evidence about accelerated arterial aging, you could look at rates of meaningful exercise across the age span. And of course, you know probably better than I do that the decline is logarithmically accelerating in how much people exercise. And one wonders whether that, in fact, could be a relationship.
Well, I think that's correct. We're just kind of like a small player in the world's pot here in the intramural program at NIH, and we don't really advertise maybe our messages in the right place, but maybe you all could take those messages and get going on that. I think it has to do with what children learn early on, what behavior they learn. And just anecdotally, I could see my children now, they're like exercise machines. They call themselves fat-burning machines, and they don't eat nearly any of the stuff that I eat. And to them, it's like a religion to have this type of lifestyle, not only for them, but for their children and what they feed them and what they teach them. And I think that's where it has to start. What you said is absolutely true, though. We did a longitudinal analysis of our VO2 max, the international standard for cardiorespiratory fitness, and we made repeated measurements over about 30 years in over 1,000 people in the BLSA. And when we looked at, what we found was the cross-sectional perspective that you get is that this decreases linearly with age. When you look at each person has his or her own control, once you get past age 40, the fall off in cardiorespiratory fitness becomes steeper and steeper. And then when we looked at, just by self-reports, physical activity, not endurance activity, but physical activity where you might perspire a little bit, that falls off exponentially as well. However, having said all that, the people in the higher levels of physical activity show the absolute level. So if you can imagine a spaghetti plot with these lines falling off with age at each decade, and then let's say there are 3 lines. One is the highest level of physical activity, one is the least. They're significantly different from each other, but the rates of fall off are the same. So it wasn't a good message for us in that regard. I'm sorry, go ahead.
Thanks. Congratulations, Ed, on another stimulating, informative, and also, I think, thought-provoking presentation. I want to just come back to George's question about whether there are things that are reduced in variability as we age. And one factor comes to mind is heart rate variability. That, without any question, goes down with age and comes down very, very tightly. So coming back to your question about stochastic processes, here's one that is not, and it's very clearly defined.
Rolf Bodmer is going to talk about that. I should have known that answer, George. I studied that for 30 years.
I was wondering, with the use of A2 receptor blockers, does that have effects on the arterial aging?
In the rat model that I showed you, it did. There are some short-term studies that were mainly done in people who have hypertension. So there have been some looks at the arterial substrate after taking receptor blockers or ACE inhibitors, and there's not a big difference between the two at the level of investigation that we're talking about. But we have a clinical trial that's achieved proof of concept to just do that, to use statins, ARBs, ACE inhibitors, or these non-enzymatic cross-link breakers. We're trying to decide which combination to choose just to do a proof of principle study. And if it works, one day we could, if we found drugs that were not very toxic, it could be like putting fluoride into the drinking water to protect your arteries with aging.
Good morning. I'm delighted to be here. I want to thank Dr. Edelberg and the members of the organizing committee for putting together such an absolutely fascinating symposium. I'm honored to be here and want to also thank the staff for making everything so beautiful. So the topic that I'm going to talk about is complementary to the topics that you've already heard this morning that you're going to be hearing later. I'm going to focus a little bit on women because it turns out that, as you saw from one of the previous slides, heart failure increases with age, and especially among women. Let's see. So, okay. Okay, so a question that plagues many of us, especially sometimes in the middle of the night, and as you've already heard from Dr. Licata, a question that we ask is, why are the hearts of older persons so vulnerable to decompensation and compromise after stress of any kind? And is this a result of the age-associated diseases, or is it a result of the cumulative changes that happen with age? So I just want to have a show of hands. How many of us think that it's a result of age-related diseases? Okay, I'd like to have a show of hands. How many of us think that it's a result of cumulative changes due to aging? Okay, all right. Yes, and how many vote for both? Okay, all right, great. All right, well, that was just so we could stretch a little bit. Okay. So you've already heard from the preceding speakers that changes that occur with cardiovascular aging in the absence of disease include changes such as decreased endothelial response, decreased coronary reserve, decreased beta-adrenergic response, and a decrease in the rate of relaxation of the muscle. This is not only cardiac, but also smooth muscle. But lest we think that everything with aging goes down, there are things that do increase as well. And these include an increase in vascular stiffness, an increase in pulse pressure, as you've heard, an increase in myocardial stiffness, and that's mostly a result of the compensatory response to the increase in vascular stiffness. And there's also an increase in the delay of conduction through the heart from the atrium to the ventricle. And this kind of summarizes pretty much what you've heard and what I think many of us have spent many hours documenting. And let's see. Okay. So on the left-hand panel is morphology, and in the top left is a scanning electron micrograph of a small artery that's been opened up. I want to call your attention to the endothelium. This is the endothelium, and you see how it's nice, smooth waves. Now, the reason these waves are here is because the artery has been opened up. In the intact animal, where the artery is actually working, these cells will be lined up along the axis of blood flow. This is a young artery, and what happens when we get older is there's marked heterogeneity in the size of these endothelial cells, as you've heard, in the thickness of the endothelium intima, and also in a heterogeneity in the direction of orientation, so that these cells and this layer is no longer smooth, this wavy pattern, but in fact they just jut out into the opening in the blood vessel, so that it's like rocks in a riverbed where they then set up eddy currents, and as the blood flow then maybe dissects a little bit underneath one of those endothelial cells, then the contents within the blood come into contact with the subintimal layer, which then sets up the whole cascade of atherosclerosis and vascular inflammation that you've heard from Dr. Licata. In the left lower hand panel is a Mason trichrome stain of cross-section of a young artery and an old artery. And I want to call your attention to the fact that there's more collagen and there's more elastin, but they're each within their own layers. And what you see is that this red, nice, lacy elastin are together, they're intact, and they work very well to convert kinetic energy when the heart squeezes into potential energy in the artery. And then during diastole, these nice elastin fibers then convert back the potential energy that they stored during systole back into kinetic energy to propel the blood forward, much like that beautiful tube that you saw in Dr. Bodmer's video that you saw. So in fact, the artery does a lot more work in mammals than in humans, but it doesn't get the credit because all this time the heart kind of claimed most of the credit, but the artery is very important and actually does propel blood. What happens with aging is that you see that the elastin fibers become frayed, they're cut, and just like the elastic waistband that we have if our abdomen becomes a little bit tight, if you would cut those elastic fibers along your waistband, the pants wouldn't be so tight. The same thing is the truth here, is that the elastin fibers are frayed, and then in between the elastin fibers is collagen deposition. So what you end up with is you end up with little pieces of elastin fibers that don't work as well. They're embedded in kind of a liquid cement. And so you can imagine then that the conversion of kinetic energy to potential energy during systole in these arteries are going to be far less efficient. And similarly, there's going to be very little conversion of potential energy stored in the artery back into kinetic energy because not much of it was ever converted in the first place. So that explains some of the changes that we've talked about. And on the right-hand side is the function. Of course, you've already heard about the echocardiographic findings that are similar in various mammalian species. This actually is from Healthy Humans, Community Dwelling. And I just call your attention to the fact that the E wave, which is early diastolic filling, is high in the young adult early and followed by a very small A wave, which is the atrial contraction part of diastolic filling. In contrast, you see that the E wave occurs later in the healthy old. These are healthy, community dwelling, free of cardiovascular disease individuals. And what you see is the A wave is actually greater than the E wave, meaning that the ventricle now really relies on that atrial contraction to implement filling. And in the lower right-hand panel is percent left ventricular filling on the y-axis and time in diastole on the x-axis, and the solid line represents a young adult, and the dotted line represents a healthy elderly. And what you see is that the young are able to manifest greater than 50% filling within the first third of diastole. In contrast, the old healthy hearts can't even hardly manifest 50% filling by the end of diastole, and the 50% of ventricular filling is actually implemented by the atrial contraction. This explains why older people are so vulnerable to increased heart rates, because diastolic time period is shortened. As a matter of fact, when your heart rate goes up and your RR interval shortens, the shortening occurs at the expense of diastole. So 90% of shortening of the RR interval comes in diastole, not systole. And so when you see that you take longer for the ventricle to relax, it takes longer for the ventricle to fill. If there's any kind of tachycardia, so when the sinus heart rate goes up, you're going to see there's going to be incomplete filling. Furthermore, if there's atrial fibrillation, there's going to be a drop in the ventricular filling, and we see a drop in cardiac output by as much as 40 to 50%, explaining why older people are so much more vulnerable to hemodynamic compromise. Okay, so we can say that based on everything we've heard this morning, and all the other studies we've heard, we know that cardiac hypertrophy tends to increase with age. We know that heart failure increases with age. And we also now know that diastolic dysfunction is more common than systolic dysfunction in the elderly. And this is the case especially in women. So the question is, why is that? Well, in part, the answer is because more oxygen and energy are needed for the heart to relax than to contract. And this is in contrast to what was long held to be or thought to be the case. Everybody thought that it took more energy to contract, but in fact, it takes more energy to relax. In addition, with age, calcium uptake by the sarcoplasmic reticulum tends to be reduced. These are some of the reasons, but certainly not all. I'm just listing a few. There's also an age-related decrease in coronary flow and arterial oxygen tension that predispose the older person or the older mammalian heart, or actually the older fly heart too, to develop hypoxia and ischemia with any kind of insult. And you already heard from the preceding speakers of some of the subcellular changes that happen with age, and they of course include the L-type calcium channel, which inactivates more slowly so that there's more transsarcolemmal calcium influx with every depolarization. And that is going to take energy to pump back out. In addition, the calcium transient is also increased in duration and sometimes also in magnitude, which also is going to take greater energy to restore. Remember that about relaxation. And the sarcoplasmic reticular calcium ATPase pump activity is also reduced. And you just heard from Dr. Bodmer about the importance of the potassium channel. And what we also know is that potassium efflux is reduced with age in the heart. And the action potential as a result of both the calcium transient increase and the reduced potassium efflux is that you have a longer action potential. Well, if you have a longer action potential, then it's going to take longer to restore. And your electromechanical transduction is going to take longer to implement. In addition, the basal production of reactive oxygen species is also increased, as we've heard, and it's good up to a point, but then, we need the reactive oxygen species for signaling, but if we have too much of it, then it's not so good. And we did a study a few years ago to look at the prevalence of heart failure in patients who were referred for study, and this is the percent of patients with preserved systolic function on the y-axis, and on the x-axis are in the category of young, we categorize those people who are under age 65, and for the category of old, we categorize that as people 65 and over. What I want to call your attention to is in the dark bar represent the women, in the striped bar represent the men. What you see is that in younger people under 65, there's quite a differential of the difference between women with preserved systolic function as opposed to men. And as the patients are older, there continues to be this gender difference, but even more marked is the percent. So it turns out that the majority, first of all, after the age of 80, the majority of people have diastolic dysfunction with heart failure in the absence of coronary disease. In addition to that, though, I want you to notice the slope of the difference in percent with preserved systolic function in the young women to the old women. You see how steep that slope is, and yet you see that there's not much of an increase in the men. And so as a matter of fact, the fact that diastolic dysfunction increases so much with age, that increase in diastolic function occurs because of the great increase in women, not so much in men. So we looked at also the prevalence of cardiac comorbidities in the patients with heart failure, and what we found was ventricular hypertrophy in those patients under 65 was about 19% in women, went up to 40% in women over 65. In men, it was about 27% in those under 65, but it only increased slightly to 30% in men. So this is a twofold difference. In addition to that, what we see was that hypertension increased by about 33%, at 36% to 49%. It increased by about 15% in men, but not so marked. And here, coronary artery disease also increased, but not at such a high rate compared to ventricular hypertrophy and hypertension. And interestingly, mitral regurg didn't increase so much, but aortic stenosis, a little bit. Diabetes, actually, not much either. So the take home here is that for women, left ventricular hypertrophy and hypertension seem to be the prevailing, predominant cardiac comorbidities associated with heart failure with preserved systolic function or preserved ejection fraction. With regards to looking at what might be risk factors for developing heart failure with preserved systolic function, what we found was that left ventricular hypertrophy had about a 2.6 adjusted odds ratio. Gender alone, just being a woman, gave you about an equivalent increased risk for developing heart failure with preserved systolic function. Hypertension gave you about a 60% increase, and age alone, just by itself, gave you about a 20% increase in risk. And interestingly here, what we saw was that diabetes, mitral regurgitation, and coronary artery disease were associated with reduced risk for heart failure due to diastolic dysfunction. Of course, the reason for this is because these patients develop systolic dysfunction even more so, so that it's not that they were so protected, it's that they became decompensated for other reasons, including the systolic dysfunction due to myocardial infarction, as well as due to dilated cardiomyopathy. So here's what we can say we know with regards to heart failure with preserved ejection fraction. We now call it ejection fraction rather than systolic function because we're realizing that systolic function isn't exactly preserved even if ejection fraction is. So what we know now is that the increased prevalence of heart failure with preserved ejection fraction with age is due mainly to the increased rate of this entity in older women. And it hardly increases in men, but mostly it is because of women. And we also know that in the elderly, for any increase in systolic blood pressure or an increase in the aortic valvular gradient for those patients with aortic valve disease, whether it's mild stenosis, moderate, or severe stenosis, women develop more heart failure for any given delta in the blood pressure or in the left ventricular systolic pressure. And we also now know that left ventricular hypertrophy results in heart failure more in women than it does in men. And we also know that older heart failure patients are undertreated and they have higher mortality. And this is especially the case for older women, as we might not be surprised to learn. Now, another finding is that older persons and now older persons without heart disease, I want to say, and patients with heart failure with preserved ejection fraction have impaired basal cardiac energetics. And you heard about this a little bit from Dr. Rabinovich and others. And the point here is that as we get older, our mitochondria are not as efficient as they used to be, and they're not able to recycle, if you will, the phosphoryl groups in our ATP molecules, so that we're not able to replete our energy as quickly and as efficiently. And it's interesting, of course, one would say, well, given all of these findings, estrogen must have a role, and we must be able to think about it, and how do we target it, and how do we fix it? So studies have shown that there are changes from estrogen on ventricular remodeling, on cardiomyocyte hypertrophy, and some of it is dose dependent, so that, for example, in the Petri dish, if you isolate cardiomyocytes and you give it a little bit of estrogen, you'll see hypertrophy. But if you give it a whole lot more estrogen, you'll see that the hypertrophy is inhibited. So then you say, oh, that's pretty cool. Maybe we should all be on estrogen. Well, the problems associated with that is, in fact, the effects are complex. And looking at older women and measuring their estrogen levels, what we're finding is that women with higher estrogen levels have a higher mortality any way you cut it, no matter what. So having a higher estrogen level when women are older is not to their benefit. So there's some complex interactions there that I think are going to need to be teased out with regards to which signaling pathway. What we do know is estrogen has many positive and potentially not so positive effects, just like you heard from Dr. Bodmer and Dr. Rabinovich, that sometimes it's a dose-dependent effect. Same thing is true with estrogen, so that it interacts with the renin-angiotensin system. And sometimes that's a good thing, sometimes that's not such a good thing. And insofar as it increases angiotensin II, that's not such a good thing. But as a matter of fact, perhaps in an acute stress situation, it would be a good thing. And we do know that there are many stories about miraculous strength in women if they have to be strong in that moment. So for example, you've heard of women lifting up the front of a car if their baby was under it. So that's a situation where they can call into acute play everything they have, and they can do tremendously strong things. But on a long-term chronic basis, then it doesn't serve them so well. Now, more recently, we're starting to understand something, which is that when women come to the ER and they say, I don't feel good, or, I'm weak, or I'm tired, and they get sent home because they're depressed. Then they go home. They come back again. Yes, I'm sorry. Okay, thank you. what we learn is that even women with normal patent coronary arteries, there's a greater increase of the development of heart failure. And we're now understanding why this is the case. This is especially the case in postmenopausal women. And this is just a beautiful slide showing myocardium, actually from animals exposed to high levels of catecholamines. Because when we're stressed, this is what we have. And so what you see is there's the myofiber disarray. Okay, this is red is for F-actin, and this green is for collagen. It increases collagen deposition between cells. And this is an electron micrograph showing contraction band necrosis here, which is where the myofiber contracts, but then the actin and myosin are locked permanently. They never disaggregate again, and the heart can't work. And so what we're finding now is that there's a very high incidence of this, what we call now stress cardiomyopathy in postmenopausal women. And it's likely that it's because of cardiovascular overactivation that is induced by emotional stress in the setting of low estrogen with reduced estrogen-mediated cardioprotection, both via direct action on the central nervous system and direct actions on the heart. So we now do understand that there is such a thing, and this is what we're looking at, is that there's an increased catecholamine cardiotoxicity that happens in women with age. So you'll see women under stress actually do have severe cardiac damage and heart failure because of this. So what can be done about it? Well, you heard about the angiotensin converting enzyme. You know that ACE inhibitor administration is helpful. You also know that angiotensin receptor blocker is helpful. In addition, calcium antagonist is also very helpful, and this is just a slide that shows that you can reverse this E to A reversal with age with a calcium blocker, and it can improve diastolic function and bring the baseline late diastolic filling into earlier diastolic filling. In addition to that, you've also already heard that exercise helps. And yes, it does, because it reduces the intracellular calcium transient, and it also reduces the duration it takes for the muscle to relax. But it's not only exercise conditioning, it also is the case for Tai Chi, it's also the case for meditation. So want to just spend one word, talk about energy modulation. You heard about already, we said that the cardiac energetics are impaired with age. This was a study where we looked at young adult and old hearts, and we looked at the ATP phosphocreatine and creatine content. And what I want to call your attention to is the fact that there's less phosphocreatine. This is baseline data in old versus young. There's also reduced creatine at baseline. However, at baseline, you notice that the ATP content is the same. But however, with stress, what happens is that you can't replete the ATP because of the reduced phosphocreatine and creatine, and this is the reason why older hearts are not able to respond to stress as well. This is also the reason why there's more of a diastolic dysfunction with age, because diastole, remember, takes more ATP molecules. Another thing to think about with regards to potential modulation and potential target and therapy is the metabolism of the medium chain fatty acids that we all use for our cardiac metabolism. And at the outside mitochondrial membrane is located the carnitine palmitoyltransferase 1. On the inside mitochondrial membrane is the carnitine palmitoyltransferase 2. And these two enzymes together help to implement the trans-mitochondrial membrane translocation shuttle of the various medium chain fatty acids then that undergo beta oxidation, and we turn it into energy. It turns out that CPT1 is reduced. It's the rate limiting step. It's reduced with age. Interestingly, and this is a study where we fed mice propionyl L-carnitine for 2 weeks, and I just want to call your attention to the fact that at baseline, 2 months and 18 months, there was a substantial decrease in CPT1 activity. However, when they were supplemented with the PLC, the carnitine, they were able to restore their CPT1 activity. But in the 28-month-old mice, supplementation with 2 weeks of propionyl L-carnitine did not result in improvement. Now, this doesn't mean that it couldn't have happened, but it does mean that perhaps the dose or perhaps the duration of supplementation may have to be longer in the older hearts. Want to just spend a minute on translational research and molecular targeting. And remember, we just mentioned the fact that it's the response to stress that changes with age and it is impaired. And part of the reason for that is because there are changes in the immediate early genes. These are the genes that turn on the entire synthetic response machinery when there's any damage or stress. And one of the genes of the immediate early gene family is the CFOS gene. And what we see is that there is a reduction in the immediate CFOS messenger RNA induction after ligation of the ascending aorta, so that it's much, much lower in the old versus young hearts. And this explains why an old heart just can't tolerate an acute increase in pressure or an acute increase in heart rate, because it can't synthesize, it can't turn on the synthetic machinery to cause there to be all of the great compensatory responses that we would need to have within the cell in order to respond. We looked at the 5' promoter region of CFOS gene and looked at 5 transcription factors, and what we found was that 4 of the 5 were either unchanged or reduced, but we found that the main transcription factor binding to the serum response element, cognate response element, was increased. And so we thought that was kind of interesting. And what we found is that, in fact, with age, normal aging hearts do have increased serum response factor. This is a protein that binds to the promoter that could and does bring about CFOS gene induction. And so the question is the age-related increase in SRF a byproduct of the aging process, or does the increased serum response factor protein itself contribute to cardiac aging? Now, the serum response factor protein is a very important protein. It's a transcription factor that's responsible for many, many, many of the muscle genes, and it's also responsible for many other signaling pathways because it's a convergence point. As a matter of fact, it's a convergence point. It's downstream of S6 kinase. It's also downstream of MAP kinase, and it's downstream of many of the other signaling pathways. So the long story made short there is that estrogen does have an effect on it, and that's an interesting question that currently is under study. Now I just want to switch gears very quickly and tell you about the new cardiovascular risk factors, both psychological and psychosocial. And what we know now is that depression, low self-efficacy, and social isolation are more powerful risk factors for poor outcome at 6 months after a myocardial infarction, so that there's higher morbidity and mortality post-MI independent of ejection fraction. These are far more powerful than just cardiac function alone. And we developed piezoelectric crystal implants in insoles that you could put inside shoes so that we could monitor somebody's stride variance. And we also developed a program where we could ask somebody to look on the computer screen, and we would ask them if they would see a flash of light and whether they could say yes or no. And then we would ask them where it was. We would ask them to press with the left finger if it was above the screen, the right index finger if it was below the screen. And what we were able to do then is we were able to also flash with these flashes of light words that were either positive for aging or negative for aging. So for example, a word that would be positive for aging would be like accomplished, wise, kind, compassionate. Negative words that one might think of with aging would be things like impaired, demented, senile, slow, or weak. And what we did is we flashed these lights on the screen for a short enough time that the seniors would see the light flash, but they could not read the word. And so what they did then is they would just say, yes, we saw the light. It's either above the midline or below. And however, we are able to recognize words faster subliminally than we are consciously, so that if we flashed a word like that, even if you don't think you could read it, it went in and you absorbed the meaning. So for seniors who underwent this, what we did is then we primed them, and then afterwards we asked them to do various things. We measured their skin conductance as a measure of stress. We measured their blood pressure as another measure of stress. And we also did mathematical and memory tests on them to see how they would perform. And this slide shows that what we found was that if we use positive priming words, we were able to see a change in skin conductance. And skin conductance, as you know, is the inverse of stress. And so if there's less, that means that they're under less stress. So this was very calming to receive positive subliminal words. So if they saw words like accomplished, wise, they did better. But on the other hand, same thing with systolic blood pressure is that when they received positive priming, it reduced their systolic blood pressure significantly. And this was all it took. This just a subliminal priming, as you might know, is extremely powerful. And the amount of reduction in the systolic blood pressure, the amount of the increase in skin conductance, decrease in skin conductance, and gait speed improved with positive priming. So the rate at which they could walk was also improved, which is physiologic now. We're talking physiology, not only indices of cardiovascular function, but outright integrated physiology. And also mathematical self-efficacy was far higher in women. I'm sorry, Stacy, I just have one more slide, but I just wanted to share this because it's so important. And so what we have here then at the end of the day is we have, of course, the whole organism, and you heard what Dr. Licata said, that it's an interaction between the very, very core information that we each carry with the rest of the environment. And insofar as we can call in to help all of the rest of the parts of the environment, we can then implement a better result. So here what we've shown is that, as a matter of fact, subliminal priming all by itself is able to increase the gait speed. I want to remind you, with this amount of gait speed improvement, it has been reported that it can be brought about after 12 weeks of exercise conditioning for 30 minutes a day for 5 days a week. So this is what we're talking about, the difference. Yes, exercise still is important, it still is necessary, but if we can call in all of the forces to help, I think we'll get a better result. And this is a role model I just wanted to share with you. It's a 101-year-old lady who went into business for herself at the age of 95. So she's entrepreneurial, which is a trait of aging. She's creative because she decided she's going to be an Avon lady. And she has very high self-esteem. As you can see, she stands tall. She has very high self-efficacy because at every house at which she makes a call, she makes a sale. She has tremendous social support because she has all this network of customers and friends, and she enjoys far better health for it. So in summary, we talked about some of the age changes that happen in the heart and in the vasculature and other places that predispose the aging organism to develop heart failure. We talked about the fact that women are at increased risk for a number of reasons, one of which could be due to postmenopausal changes, but other things also play into that. On the other hand, she is still very resilient, she's very strong, and if we can call in to help all these other attendant potential powerful forces, we can help to rescue her as well. Thank you so much. I appreciate your attention.
I think we have time for a question. George?
I've got a hypothesis. Maybe you could test in Arkansas. So with all the stress that the women have, is it correlated with their husbands retiring and hanging around the house? So, which means that the best thing we can do is raise the retirement age.
Yeah. So Dr. Martin raises a very fascinating question. I'll just share with you a very quick vignette. The other day I was in a fast food vendor place, waiting in line just to get something, you know, like coffee or something. And I overheard these two ladies. This was on a Sunday morning. They were sitting there. This one lady laments to the other lady, "I cannot tell you how horrible it is. He is constantly underfoot. I don't know what to do. And every time I try to walk into the next room or I want to go to the market, he says, 'Where are you going? I want to come with you.' And she says, 'I just can't take it anymore.' And so the other lady says, 'Well, you know, I have to tell you, my husband actually had early retirement. So I've been putting up with this for the last 10 years, but you'll get used to it.' So what Dr. Martin says is partly interesting in that we all have to make adjustments in life. And when we're used to doing certain things, and then when things are different for us for a variety of reasons, some of us actually do have to call in all of our other compensatory mechanisms. But I want to come back to the stress cardiomyopathy thing, because this is what affects older women. This is what affects postmenopausal women, is when they're under some kind of stress. I just had a patient last week. Her brother died, and she went out to the backyard to water her lawn, and all of a sudden she had this terrible crushing pain. They rushed her to the hospital. She had a myocardial infarction, but they looked at her coronary arteries. They were completely clean and open. So there is an absolute case of a stress cardiomyopathy in the making because of catecholamine cardiotoxicity. Because when we have a higher estrogen level, it protects us from this catecholamine stimulus, both to the brain and to the heart. But when the estrogen level starts to go down, we need to call in other defense mechanisms. And these include, for example, blockade of the angiotensin receptor, renal angiotensin system. It calls into the defense of, for example, the increased intracellular calcium transient. So, and or as well, our own perceived stress. And I think these are the things that we need to think about. So thank you, George.
Thank you very much indeed for inviting me to this symposium. And I come from the vascular biology field. I found autopsy, we had enough of it. I went into imaging, MRI, and I'm a clinician who sees patients. So therefore, I will give you a perspective of how I see the topic that has been assigned to me today, trying to give a view of 4 different questions. I'm going to talk about aging, which what we have to do is to promote cardiovascular health, and you will see in a moment why I wrote 2009 and 2019. Now, the atherothrombotic disease is the subject I'm discussing, and I'd like, first of all, to talk about 2 main challenges that we have today. Challenge number 1, the number of people in terms of prevalence over the age of 65 in the United States will increase from 12% to 19% in 2030. And this means disease. What kind of disease? Atherothrombosis and its consequences, which is cardiac failure. 80% of the patients with cardiac failure is due to atherosclerotic disease. So that's the first issue. But what about worldwide? We need to have an open mind here. This is the population worldwide. This is 2000. This is what is projected 2050. This is a real issue that there is no need to give you numbers. Just look at the slide. This just came out from the WHO. Second challenge. Paradoxically, despite their increased risk of cardiac events, elderly patients are less likely to receive appropriate therapy. It's always the same thing. Benefits versus risks. One feels, well, it's not necessary, he's going to have a problem, and you just do not treat the patient. When in fact it's all the contrary. If you look at cholesterol, for example, you have the same decrease in mortality in a young person than in an older person, but since the denominator is much larger, the impact is much more significant if you treat the elderly. So this is one issue that we have a second challenge. We do not treat these people. But let me tell you what's going on in the world. If we talk about what really causes the disease that we are talking about today, look, hypertension, number 1. Just came from the WHO, 2002, and I have the data now in 2008, is very similar. This is the problem. Hypertension, tobacco, cholesterol, lack of exercise, diabetes. So what we are really dealing here is with a huge problem that if we are going to talk about research, we have to do zebrafish, of course, mouse, but humans, because if we don't cut what is going on here, we are talking to the skies. And this is a reality that I want you to be very aware of what is really happening today. Having said that, let me now go into point number 2. And it's an interesting point that has been already discussed. Is biologically, is vascular aging similar to atherosclerotic disease? And my answer is no. I'm sorry to say you that, coming from the area of imaging, as I'm going to be discussing in a moment. First of all, this was beautifully presented already. Aging has been associated with changes in the arterial system, are structural changes, collagen, elastin fragmentation, less elastin, and then there is endothelial dysfunction. And this is an increase in systolic pressure due to the stiffness. And certainly at the very end, if you have a high blood pressure, you may affect the brain and the kidney, and if you have a low diastolic pressure, you may affect coronary perfusion. So there is no question that the problem of aging has significant implications, particularly in the differential blood pressure and its consequences in the end organs. But be sure we don't confuse these with atherosclerotic disease. And this is what I'm going to be discussing in a moment. Now, what is aging? And I have actually not much to add to what has been said already. And basically, there are a number of stresses, oxidative stress, as has been shown, that may affect the cell, and the cell may die in 2 different ways. One is an apoptotic pathway, and the other is cell cycle arrest. Now, what is interesting is that during the apoptotic phenomena, these cells release substances, which is a protein expression, which tend to be toxic. Be sure we remember this for a moment, because this has significant implications in the vessel wall. If we go now into more detail, here you have the telomere. The telomere is where the Nobel laureates came in the last 3 days. These are the people who really describe the impact of the telomerase into breaking this telomere and leading to cardiac tumors. And the question is, how can you modify this pathway in the future? But this happened actually also in the vascular region, and that is this breaking of this particular component may lead to the cellular senescence that we said in the arterial wall, and this is either through the apoptotic phenomena or to cell death. And then there are other aspects, as we already discussed, independent of the telomere, which is, for example, oxygen radicals and so forth, which may lead to the similar phenomena. But what we are really discussing here is aging of the vascular wall. And if you go into more detail, what is becoming very exciting today is this is all regulated by these 7 enzymes, the third family, which is quite fascinating, whether this can be modified in the future. They tend to be actually regulators in the favorable way, like trying to prevent the process of vascular senescence. Now, what I'd like to show you here is very important. Over the last 5 years, we have been doing MRI of the arteries of people of different ages. And I only want to give you a summary, which is actually, this comes from pathology. Our summary is all in the literature with Zahir Fayyad. We see progressive intimal thickening, no connection with focal atherothrombotic disease. And one of the most interesting issues today, everybody talks about intimal medial thickening as being a parameter of atherothrombotic disease. This is being challenged, and we have now 6,000 people, which I will show you in a moment, in which we have done 8 imaging technologies in following the arterial system. And we are learning a lot that is not coming from autopsy, it's coming from longitudinal studies. So we have to distinguish what it is vascular senescence, which is a progressive process, which I believe may in a way help or trigger the focal atherothrombotic disease we will discuss in a moment, but I think are 2 independent phenomena, which is this is actually very diffuse. Having said that, let me now go into atherothrombotic disease. Is this a focal disease? Certainly when there is a blood clot in the system, you can say it's focal because it is in a focus, but this is a incredibly diffuse disease, as you will see now with imaging. But let's just touch into what age has to do with the atherothrombotic process. And I will say from the very beginning what has been said already, and that is the incidence, the prevalence, and the mortality due to coronary artery disease increases with age. I don't think there is any question about it. The question is why. And there are three possibilities. One possibility is that the risk factors keep adding over time, more hypertension, more than before. The other is the cumulative same risk factor that is present over life. And the other is the substrate, what I am presenting to you right now, the senescence of the vascular system. I'm sure it's not too helpful when you have other stressors that are ready to cause the atherothrombotic process. My view is, I don't think anybody has the answer, but probably the answer is a combination of the three. And if I had to put my finger on, there I see the senescence is probably very important as a background to where the problem is, as you will see in a moment. Let me now continue by saying why I'm presenting this slide. I'm going to be presenting three phases of the atherosclerotic process in which age is critical. The first one is endothelial damage due to the conventional risk factors. We all know this. And we also know that progenitor cells today unequivocally find a fantastic home to substitute the damaged endothelium by the risk factors. I don't think anybody questions this. This is just experiment where you do in an animal model, in the rat, you do angioplasty, and then you label the progenitor cells from the animal with iron, and then at autopsy you can see by immunofluorescence how the high concentration of cells are in the place that is de-endothelialized, but you can follow this in vivo also with MRI. There's a lot of information, and that is if you want homing for progenital cells in a damaged heart, the best homing is the damaged endothelium by far. I believe much more than the myocardial damage in itself. Having said that, here comes the issue of age. And the issue of age is that actually these progenitor cells, which are endothelial progenitor cells, actually with age, they deteriorate. And this is actually very well proven. So what you have is endothelial damage with the risk factors. You have hypertension, and you don't have enough progenital endothelial cells to go there. But at the same time, they are dysfunctional. And one of the interesting issues here, it appears that the same risk factors that affect the vessel wall affect also the structure of the progenitor endothelial cells, except one thing, which is exercise, which stimulates them. And I just want to point out that this is a very hot issue. The issue of progenitor, the age of the progenitor cells that try to repair tissue. And one of the problems with age is that what I mentioned to you before, this telomere is much more activated, the breakage, with age than in the young population. And this is obviously leads to the senescence that I mentioned to you before, but if we are dealing now with progenitor cells, what we are doing is they are incapable to substitute the damaged area, and this is something that goes on with age. And I think this is very important because maybe when we talk about senescence or this vasculopathy that is generalized with age, I wonder, and I don't have the answer, whether some of the endothelial that is damaged and leads to intimal thickening might be populated by these cells, and then what happens is the pool is too small or too low, and so forth. This is something to take into account. And what I think is very interesting in the field is now this concept of old progenitor cells is beginning to be looked at on many diseases, not just vascular disease, but also diseases in many other organs. And I will talk about the brain in a minute. And what is really evolving now, and the data is being very strong, is not only what you commented on before about exercise that is good for your vasculature. it appears is very good also for the well-being of the so-called progenitor cells. And the data in animal models in the last year or so is quite convincing of that. So this is something that we have to take into account as we try to repair a damaged endothelium, which is what I'm talking about. The second part is interesting. I just talked about progenitor cells and the problem with age. Here there is another issue. And the issue here is the atherosclerotic disease, contrary to the senescence, is a reaction of the artery to a foreign body. What appears to attract the progenitor cells are apoptotic cells that are endothelial cells that are dying, and they are recognized as foreign bodies. Here is the same thing. This vasa vasorum, as a second stage, tries to take the cholesterol deposition out from the artery here. But these vasa vasorum are very primitive, and they leak red cells, which obviously is a foreign body that attracts inflammation. And the problem of this inflammation is he is very active and may rupture, the enzymes from these monocytes may rupture the internal elastic lamina, a site of taking care of the foreign body, and this is what makes a plaque vulnerable. What would be the answer to this? The answer to the first question is, how do we learn more and more about progenitor cell biology to really, at the molecular level and cellular level, to take care of the damaged endothelium? The question here would be, can we lower LDL in this vasa vasorum significantly and by a gradient of concentration to take this deposition out? And we have done all of this with imaging, and we have now 100 patients, which actually 75 were diabetics, which they had with MRI. You could see the deposition in the arteries, this is in the carotids or in the aorta, and we gave simvastatin to 50 individuals and atorvastatin to 50, and in a period of a year to 2 years, this lipid material goes out. And we believe that that's critical when we give statins, as we favor a defense mechanism, which is the pulling of the lipid material that enter into the vessel wall because of the damage of the endothelium. Now, this is actually very fascinating because now when we go to the elderly, we face the problem. What are we doing with LDL? And there's no much care to the elderly. That's the problem when I'm presenting to you the biology. When in fact the best data, when it has been done, when it has been given to people over age of 70, they drop mortality and so forth by giving statins with an LDL of less than 70. This is the data. So here, what I'm presenting to you is a biological phenomena in which nobody cares when you reach age 65. I'm putting things in an extreme just to understand is a problem. So to me, this is a challenge. We talk about the elderly, we can talk about anything. We are not treating them. And here's an example. And then the other issue is cost effectiveness. This data is just evolved. I'm sorry you don't see this in the slide. Lower you are here, more cost effective you are. One of the most cost effective things is to treat the elderly, even rather than the younger, with the statins, by the impact that they have. This is very fascinating. It's the opposite of what one might think, and that is maybe it's not worth it, when in fact is the most cost-effective thing that we can do, based on what I am presenting right now. And this comes, the third issue, which also has to do with age, and this is another defense mechanism. The lipid material entered into the vessel wall because of the damage of the endothelium. And now these monocytes recognize. But the problem is this inflammation has one issue. These monocytes very easily commit suicide. They become apoptotic because they cannot do the job. And now comes what I told you at the beginning. During the apoptotic phenomena, they actually release toxic substances. The most important is tissue factor. And tissue factor is the factor that initiates the clotting system. So what I really define for you is a myocardial infarction in 2009, which is the failure of the whole defense mechanisms. The endothelium is not properly covered by progenitor cells with the risk factors, and lipid material enter into the vessel wall. This hemorrhage occurs because we didn't drop LDL far enough, and the inflammation wins, and takes care of the red cells, but also the internal elastic lamina that breaks. And finally, once this breaks, you have a plaque that we call vulnerable. This plaque then may open into the lumen, and the blood that circulates find the toxic material as the failure of the third mechanism. Why I'm presenting the third, what this has to do with age, the third one, has a lot to do with age, as you will see in a moment. The issue is this apoptotic phenomena, actually, we now can recognize, certainly in the carotid arteries, with imaging. But one of the things that is becoming very clear is the so-called toll-like receptor is very important and can be modified with age in going against you. And this is one of the problems that has been identified. Without going into the details of the slide, how the toll-like receptor plays a very important role in the issue of the plaque, I want to present to you that if this monocyte that tries to recognize the lipid material and actually takes it, the bad cholesterol, and delivers to HDL, why we don't increase HDL ApoA1? And here's where the issue of toll-like receptor and age come into the play. Because what basically happens here is if we increase ApoA1 in the blood of a rabbit model in which the process has stopped with apoptotic phenomena, the whole thing gets reversible. What ApoA1 does, very fascinating, what ApoA1 does, on one hand, it enhances these transporters of the oxidized LDL. But what it does also, it blocks toll-like receptor, which is very much age-dependent. Older you are, the toll-like receptor, more active it is. And as the cells die in apoptotic phenomena, it reacts bringing more cells in and is getting more and more toxic material. So what ApoA1 does, it blocks completely the toll-like receptor, which is very sensitive with age, and this allows the monocytes to do the job because the HDL is high, because the ApoA1 and the process is actually stopped. So this is an issue that is very important, which we have documented in the animal model, and also with MRI, which is fascinating. This was published very recently with Juan Badimon. In the rabbit model, when we give ApoA1, we decrease significantly the inflammatory process, and we have not identified toll-like receptor, but certainly the transporters are enhanced and tissue factor is not generated. What I'm really saying to you is that when we talk about age, we really have to go into the biology of the plaque and trying to understand that age has significant implications in enhancing the phenomena of what we now call focal atherothrombotic disease. And I presented to you the three aspects of defense that really tend to fail with age. And this is something very important that we have to take care of. But I talk about the last phenomena, which is basically the inflammatory phenomena that goes into where the lipid gets deposited and may release tissue factor. Now let's assume that the plaque breaks and there is a blood clot. What do you do, and what age has to do with this phenomena? I'm not going into much details, but to tell you that the most common cause of hemorrhage is age. And this is in all the studies. That is, here we have a problem. When you treat acutely a patient with whatever drugs you do, thrombolytic therapy and so forth, during the acute phase of myocardial infarction. But what is fascinating, things are changing very rapidly. We have two drugs presented in the last meeting of the European Society, which for whatever miracle it is, they do not cause the bleeding that we expected. And the reason is because they are very reversible. These are patients with acute coronary disease that are giving an inhibitor like aspirin. In this case, the name of the inhibitor is ticagrelor. It blocks a pathway of the platelet, and interestingly, is the best drug available today, very rapidly reversible, and bleeding did not increase as age increased in these patients. We are talking about 1,000 people. And the second drug that showed the same thing is a direct thrombin inhibitor, like an anticoagulant that you see here, dabigatran. This was given chronically in patients with atrial fibrillation. And without going into the details, is another miracle. That is, this drug does not cause the bleeding in the elderly, that was found before with oral anticoagulants. What I'm saying to you, very exciting, atherosclerotic disease is the disease that kills people, but the final killer is a clot that you have to take care of. And older you are, more trouble you have with the issue of bleeding, and this is now moving very rapidly. And I'm just going to finish by making a couple of points. The first point is, after I said the importance of atherothrombotic disease versus vascular aging, is what about vascular disease in the heart versus vascular disease in the brain? And I just want to point out one thing. Why the kidneys are affected at the same time that the heart if you develop hypertension? Why the kidney disease has the same risk factors in the heart? Therefore, let's move from the heart into the kidneys. But if we move into the kidneys, why we don't move into the brain? And this is what is happening at the present time, where there is very good evidence that cognitive function, degenerative brain disease, has a lot to do with vascular disease that we are discussing today here. The thing is, it's nice that you say the heart as an independent organ. I'm sorry, I would change the topic. The heart cannot be viewed as an independent organ if you talk about the vascular system, because the problem here is this is the same process in the heart than in the brain. And I have to tell you, all the studies that are showing now, as blood pressure goes up, cognitive function goes down. This is all the data that has evolved in the last few years, how hypertension can affect cognitive function and how hypotension can affect cognitive function. And this is data that all of us who deal in the intensive care units know very well with the surgical patients. You drop the cardiac output and the patient doesn't know what he's talking about. Here is data. Lower is the cardiac output, more is the perfusion of the brain being affected. So to say I'm going to treat blood pressure bringing 120 systolic on an individual age 75, I'm not entirely sure we know what we are doing. Blood pressure being high is a problem. Blood pressure being too low is a problem. And I'm just presenting this to you because this has to be investigated. This is something we deal every day and has to do with the age of the patient. And this is just an outline that your cognitive function can be affected by a stroke, but can be affected by a small vessel disease. And here comes the issue of Alzheimer's, which you know that the present concept of Alzheimer's is two phenomena. One, the combination of age, hypertension, and amyloid. This may close the vasculature, the small vasculature, which affect the neurons, and this may affect the neurons directly. This is all the people who are working on Alzheimer's today are dealing with the same phenomena that you guys are discussing in this room. The vascular component with the risk factors we talk is extremely important in this disease, and therefore is something that we have to pay attention to. And now comes my final comment, which is actually, you will say, maybe is a paradox. I told you a moment ago that atherothrombotic disease is a focal disease. Senescence of the vascular system is diffuse. But I said, when I say it's focal, means the event is focal. Is the disease focal? These are all the risk factors that we deal today in the adults, are about 7, and you know all of them. This is a study of 6,000 people with more than 2 risk factors in this country, in which we did 8 imaging technologies, carotids with 3D, calcification of the coronaries, blood pressure ratios in the legs, ultrasound of the abdominal aorta. And if one of these techniques was positive for disease, they had a high risk factor profile. We did MRI, CT, and in some of them positive emission tomography for inflammation. I cannot go into presentation of the data to you, but I want to give you an example which gives the message. This is 3D, which actually it shows a lesion here, which you would say in the carotid artery is 1 millimeter, and you say no problem, less than 30% obstructive. No problem, except when you do positive emission tomography, which is looking at the inflammation I presented to you today, 3 types of inflammation, look what you see. The carotids are all yellow. This is inflammation with very sophisticated technology. And then you go to the legs. This individual had no symptom, more than 2 risk factors. And what we are seeing is the disease regionally is diffuse, but systemically is also diffuse. And this deposition of inflammation has very little to do with the so-called senescence of the vasculature, which is very much collagen, less elastin, and calcification. This is all inflammatory. And this is why there are two different diseases. What I believe is the senescence may contribute to this phenomena, and this is something that has to be investigated. And I finish by saying, every time I talk about these things, this is nice to go with a mobile unit. This is all research. We try to find the best technology and the cheapest that can tell people who are developing the disease before it's apparent. But what about, are you going to Rwanda with a mobile unit? You can't. And 4/6 of the world are middle or low class, low economy. Therefore, you have to start putting your brain on gear. And this is the reality of the slide that I want you to really look at. High economy country, middle economy countries, low economy countries. Look at cardiovascular disease. Number one killer, 2004, 2005, 2030 will increase. But look in middle income countries. And now look in the low income countries. Do you think that what we are discussing today is not affecting the poor countries? They have the same amount of disease today, chronic diseases that we have. Why is that? They keep the food with salt, hypertension and stroke. People eat cheap, carbohydrates and obesity and diabetes. And the companies, the tobacco companies are there and nobody knows they are there. They are absolutely infiltrated and the young people are smoking. And I could present to you data that we are obtaining now in Rwanda, which is amazing. You would never believe that. So that to me are challenges that we really have to pay attention to. And I just want to say you that, lucky or not, I am the chair of a committee of the new administration, which with the IOM, we are going to have a document next year, which is how we solve the problems that I presented to you today. And that is, what in the world can we do on an epidemic, as I show you in the slide, that is evolving? I call this research. This is research. And you see 2019 is because we are looking at the next 10 years. This is why I began from the very beginning in giving a perspective of where the issues are. And I just finished by saying to you that I think we have a challenge here, and the research has to be at all levels. Has to be at the molecular level, at the cellular level, at the epidemiological level, and so forth. And keep in mind that we are in a part of the world which is high economy. 4/6 of the world are not. And therefore, we need to keep a very, we have to really have our conscience very clear of what we should do. Well, thank you very much for your attention.
Thanks a lot, Jay. I particularly want to thank Jay and Roger for the invitation to speak here, and also for the organizer for this wonderful conference. I really learned a lot about aging. And I have to say, my disclosure slide actually is not about what companies that I actually affiliate, but for the fact that my lab actually does not do aging in any specific way. But I hope with the talk that I'm going to give you is the sense that what we do actually will have an important implication for aging, in particular the idea of how we might regenerate a diseased heart. So, as most people here know, that currently heart failure is a rising epidemic, and that the kinds of therapy that we have kind of comes in two very broad spectrum. And on one hand, you have sort of the tried and true drug therapy with beta blocker and ACE inhibitor. I'm just trying to see if. All right, the laser pointer's gone out. Well, there we go. Another one. So the drug therapy, as most of you know, has been the bread and butter of heart failure therapy. And then on the extreme end, the invasive part, a cardiac transplant for patients who are at a point where the disease has progressed, but obviously there are not enough hearts to go around. So a lot of people have been looking at efforts to try to fill the gap between these two different approaches. So some of the new ones that has come on recently, like natriuretic peptide using different peptide-based therapy, and Hazel has described very nice effort using peptide, and that potentially will be providing significant benefit in patient with heart failure. But on the mechanical end, which I think has recently really has taken hold, where a lot of people are either using mechanical device like ventricular assist device or using synchronization therapy with biventricular pacing. These have really have
sort of made the heart failure therapy advance in a much more invasive and mechanical direction than I think most people would have liked. And the gap that we're trying to fill is what, as Roger mentioned, about using things like gene therapy, and then also there's been a lot of interest in using cell-based therapy or a combination of genes and cells. But obviously, right now, none of these are standard of care, and that much testing is needed. So I want to give you a bit of a tour of where we've been in cell-based therapy because of the interest that many people have had. In the sense of the kinds of cells that people have been looking in transplanting, about 10 years ago, we started with the use of skeletal myoblasts, and I'll talk about that in more detail. But more recently, a number of different populations, like either endogenous progenitor cells or using bone marrow as a source for potential cardiac regeneration. And that now has had some interesting results from clinical studies. But much further in the horizon, the use of embryonic stem cell or induced pluripotent stem cell, which I'll talk about later on, are also creating a lot of interest and excitement. So how this first started in recent era, I'm sure probably in beyond sort of the last decade, people have considered the possibility of using cells. But Doris Taylor, while she was at Duke, had started the study of isolating skeletal myoblasts, working with Bill Krause, who was a real skeletal physiologist. And what they had looked at was the possibility that if you isolate skeletal myoblasts, which people have isolated characterized from adult skeletal muscle, and transplant them into an injured heart, could you get the myocyte replacement that you would like, and perhaps some remodeling and some other benefit along with it. So what they've shown is that you get a lot of scars in the injured heart without transplanted cells, but you get this very large area of engraftment from skeletal myoblasts, which look very impressive, and they also shown there was significant functional improvement. So from that study now almost 10, or actually more than 10 years ago, there's been a series of clinical trials that have led to looking at the possibility of doing this in human, and a lot of small trials that have suggested benefit, but the one that really has come along to trying to get a more definitive study was done by Philomena Shea from France. And what they have shown in this study called the MAGIC trial is infusion of 2 different graded doses of skeletal myoblasts did not really significantly benefit us. So this is comparing with untreated versus low or high dose. The functional improvement was really just not there. They saw there may be some potential improvement in ventricular remodeling, and what they concluded was that myoblast injection in patients with depressed LV function, this was done at the time when patients are undergoing cardiac bypass, did not significantly improve their ventricular function. But they thought what was interesting, there may be some improvement in the remodeling, but what was the real Achilles heel of using skeletal myoblasts was the arrhythmic event. In essence, all the patients had to have been implanted with an ICD device because there was what was considered as being electrical incongruity between the skeletal myoblast-derived cardiomyocyte from the existing myocyte. So, but nevertheless, I think as a first foray, it suggests that there are some benefit related to injecting cells that probably is there and maybe can be improved upon. So subsequently, investigators, in this case, Piero and Versa, have looked at the possibility in animal models of injecting bone marrow cells. And now I know most of you have heard a lot about different bone marrow studies. This study that really triggered the interest in using bone marrow cell that Piero had shown was that he had demonstrated in these area of large infarct, if you inject these GFP fluorescent protein tagged cells, they can eventually differentiate into sarcomeric actinin, here stained in red, cardiomyocytes after transplant. And they've also further demonstrated in animals that you can get a reduction of the left ventricular end diastolic pressure, as well as improvement in the LV-DPDT, or the fourth generation. So, you know, from the experimental studies, this quickly caught on, and that number of clinical trials since then, over the last 7 or 8 years, have taken on hold. And the one that most people now cite came from in 2006, 3 studies that were published back to back in New England, well, 2 in New England, 1 in Lancet, have basically shown that in patients in the acute setting, ST elevation MI, transfer of autologous bone marrow cell in the Belgium study did not show significant improvement, and In the Swedish studies, they shown also there was no significant improvement. And the only one that showed improvement came from the German group by Andrew Zeihar. And they shown that it appears to be associated with a moderate, but it was statistically significant improvement after a short period of 3 months. And they have subsequently gone on to show that there is also some modest improvement after a year. So there's the appearance that there's something going on with bone marrow cell transplant. And the part that I think that has come into a lot of controversy was whether the cells actually differentiate into cardiomyocytes. So the initial study in animal from Piero suggests that there was cardiomyogenesis, but a number of studies subsequent to that have shown that there was no evidence, particularly the one came from Chuck Murray at University of Washington, as well as from Stanford group, have also shown the absence of cardiomyogenesis. So I think what probably the consensus, at least as much of a consensus you can get with a bunch of cardiologists, is that the amount of cardiomyogenesis is rather minimal, but there are other effects, particularly paracrine secreted factors that these cells must carry at the time when the cells have been transplanted. So a number of efforts are going on in looking at what the paracrine factors are, and potentially in the end, we may not actually need the cells, at least bone marrow cells, and if we can identify what the actual secreted factor. So in the midst of all these various different efforts in looking at cell-based therapy, we decided to take a step back in looking at what exactly is the cardiomyogenic process that is required for you to really generate a truly functional cardiomyocyte, whether or not you're deriving it out of bone marrow cells, out of other adult stem cell population, out of adult cardiac stem cell. You know, the process in which the cells eventually give rise to cardiomyocyte has to be reasonably similar or conserved. So the ideas that we began to look at was that when you go through from the entire lifespan, from embryonic to adult and eventually to aging, the ability of the heart to repair and regenerate progressively decline while the number of injury and insult progressively increase. And the fact that the number of cardiac progenitor cells that are still present within the heart probably is part of the reason for this decreased ability to repair and regenerate. We begin to look at the possibility of this parallel relationship of the presence of progenitor cell with the ability to regenerate heart. So what we first then looked at is we went back to the source. So where are the progenitor cells during development, and what are the characteristics of those cells? So in the studies that we published now 3 years ago, which I won't go into detail, is that we use a genetic labeling approach where we tag the early heart-forming cells driven by a NKX2.5, which is a cardiac restricted transcription factor, and label those cell population with enhanced green fluorescent protein. And we show that in the very early developing heart cells, these cells turn green, and we were able to subsequently show they retain multi-lineage differentiation capacity into not just cardiomyocyte, but smooth muscle cells, and with just a little bit of endothelial capacity. And we summarize that in these early development, a early precursor by the expression of NKX2.5 at that stage is able to give rise to cells that phenotypically represent atrioventricular myocyte, some of which also generate conduction cells, as well in small population vascular smooth muscle cells. So this allows us to set the template for trying to understand what genes and factors are involved in the process of cardiogenesis as a way to look to see how we might be able to adopt that in other settings. And concurrent and also following our studies, other groups have also shown that cells with capacity for multilineage differentiation can be found from other sources. So Ken Chen's group has shown that islet-1 positive cell from a second heart field, so another portion that also give rise to the developing heart, particularly the right ventricle and the outflow tract, also undergoes multilineage differentiation. And what was more surprising was that two groups, and we were part of the effort that Bill Pugh from Children's have shown, is that the cells that lines the outer surface of the heart, called the epicardial cells, actually also retains the ability to become cardiomyocyte through early embryonic development. And a lot of efforts are now looking at the possibility of whether the epicardial cell may actually represent the population that could undergo regeneration in the adult heart. So this is the only diagram of development that I'm going to show you, since I know most people are probably not developmental biologists. But in the early forming heart, as I've shown in the GFP picture, the first heart field represented in green by the expression NKX 2.5 and in the blue by the expression of islet 1 that I mentioned in the second heart field. The two fields undergo a lot of morphogenetic changes and regulate the formation of the developed heart. And that from this point out, a lot isn't really unknown about what happened to these early forming precursors as they go all the way out into the adult heart. And a number of groups have shown that the expression of either c-KIT or SCA1 or Psi population by ABC transporter represent a stem-like population in the adult heart. And the interest had been to trying to understand exactly what is the biological relationship between these developing precursor to the adult heart. Because if you could really follow and track all the way from the developmental stages all the way out into the adult, you might be able to see which population are really the ones you're interested in expanding and inducing to undergo cardiomyogenic process. So we decided to look at that question using the GFP mouse that I just mentioned. And by using flow cytometry for the GFP positive cells, what we did is we went all the way from the early embryonic days. You can see the embryonic day 8 to 12 in the mouse. There's quite a high level of GFP positive cells expressed at that stage. And then progressively towards mid to late gestation, that population is almost completely gone, but not quite. And what was interesting to us is that during the very first 3 weeks after birth, there's actually a re-expansion of that GFP population in the postnatal heart. And we were quite intrigued by this population because at least up to this point, no one has really quite made the link between the developmental precursor with anything that has been found in the postnatal heart. So we went further to try to describe this population some more. So this population are particularly located along the outer surface, which, as I mentioned about the epicardial derivative, these cells are likely the remnant population from those early epicardial precursor, and then they progressively march into the myocardium as they undergo their initial wave of development during the neonatal window. But the population of this diminished quite a lot. And what was interesting is that this mimics a lot in the recent study that has been looking at exactly how much cardiomyocyte renewal and turnover actually exists in the adult heart. So this was a study that published by Jonas Friesen just a few months ago in Science. And what they did is to utilize something which was quite unfortunate back in the 50s. So the nuclear fallout back in the 50s had generated such an enormous amount of carbon-14 in the atmosphere. And what you can see is that during those nuclear testing era, there was this rise in C14 level, which then with the nuclear testing ban, there was subsequently a decline in the amount of C14. But the half-life of C14 is so long, it's somewhere in the 2,500 years, that basically whatever that gets incorporated into the body stays there as long as the cell does not turn over. And then so what they did is to utilize essentially sort of the anthropologist approach to looking at cell turnover in the human body, is to look to see on any organ, on any cell type, what exactly is the content of the carbon-14, and then measure that all the way back to track with what level it corresponds to in the atmosphere. And then if you see the differential between the person's biological birth year and the year that corresponds to the tissue sample, they give you the representation to how much turnover that there has been in that particular organ or that cell. So what they found in this very unique way of looking at it, completely unbiased, is that there's about 1% renewal in a young person, like 25-year-old, and in humans. And that progressively declined with age to about 0.5% in the 70-year-old. And cumulatively, what this means is that about 60% of the heart that you have by the time you're in the 70, 80 years old, the myocytes came from the time when you were born. So there's really very little normal turnover. And that's actually consistent in the mouse model that we have with looking at the progenitor cells, is that there's this initial burst right after birth, and there is a significant decline thereafter. But that's during normal homeostasis. What happened during injury? So, Rich Lee had a very nice study from 2 years ago looking at what cardiac regenerative capacity there are in the injured heart. And what they found, quite surprisingly, is that there is apparently a stem or precursor-like population that replenishes cardiomyocyte in the area of injury. And this is distinctly different from what the normal turnover that Jonas Friesen has shown. So the interest then is to try and figure out exactly who are those repopulating progenitor and stem cells. So we used the mouse that I just described in an injury model in trying to see if you look at these very early stage neonatal mice and then progressively further out, do these mice actually reactivate that diminishing population? And I had a really talented postdoc who had taken the challenge of trying to perform a LAD ligation in a 1-week-old mouse, which is about the tip of your pinky, and it was pretty. The whole mouse is the size of the tip of your pinky, and he had done a really nice job, and we were able to see exactly what happened if you go from something that was fairly easy, like a 6-month-old mouse, and ligate the coronary. You see an expansion of the GFP with the MI. And down to 6 weeks, 3 weeks, and 1 week. And there was so much progenitor that were there at the time, so you don't really get an extra boost by having the injury. But nevertheless, the ability to re-expand after injury was encouraging, and sort of led us then to look at, do these cells actually contribute into new cardiomyocyte formation? So then we had to generate another mouse that allowed us to look at that. And in this case, we used a genetic labeling approach using the Cre recombinase system, where we had a mouse that is driven by an NKX2-5 enhancer that I mentioned that was driving the GFP, except in this case it drives a Cre recombinase. And when you cross with the LAGC reporter mouse from the ROSA26 locus, so this mouse, if the Cre is expressed, there is a stopper cassette that's excised, and then the LAGC turns on, and you can see the cells from the time that the Cre is expressed and all the subsequent progeny, because the excision comes within the genome. And so by using this system, we then label the mouse at the time of injury, so when the progenitors are re-expanded, and then we follow through for the remainder period up until 12 weeks after. So now, in this study, there are a lot of different people would question things like, well, are you reactivating cardiomyocyte? Are there already some endogenous cells, and so on and so forth? So we had to do a lot of controls to be sure. So one control is to be sure there was no labeling with this inducible mouse, and you can see before we induce injury, there was no LAGZ signal at all. Represent the baseline is completely silent. And then without the injury stimuli, which we knew there was very little progenitor in that state, we follow the mouse for 12 weeks without injury, and again, you don't see much of any labeling at all of cells. Whereas, and also with the LAGZ only, in this case without the Cre inducer, so we don't see any spurious blue cells that is induced after injury, you don't see any of that. So what happens then if you then have both the Cre mouse and the injury with the reporter? So what's interesting is that at 1 week, there's hardly any labeling, but I'll show you there's actually a little bit right along the edges, just like what I mentioned before. Okay. And progressively with the recovery phase, you can see from 3 to 6 to 12 weeks, a lot of cells now have turned blue, represent the descendants from the original progenitor cell. So we think we have now finally captured a population of regenerating progenitors that gives rise to myocardial repair, and that these cells lies along towards the edges of the heart, and they eventually differentiate and are stain positive for alpha sarcomeric actinin for cardiomyocytes within the heart. But the amount is not that much. So we were able to capture about 4% of the myocardial through serial section that represented the regenerating cells. And obviously that's really not enough for the billion cells that we lose during one large myocardial infarction. So we need other strategies to enhance this. So we recently have teamed up with Stan Shaw at MGH Center for Systems Biology to look at the possibility of taking the cells on a screen and looking for pharmacological expansion of this population. So by isolating the GFP cells, put on a 384-well array, and through pin transfer with a large-scale high-throughput compound assay, and then measure for the expansion of the cells and looking at high-content screen for progenitor characteristic, we're interested in finding compounds that helps to expand the population. So this is a very early data we have. where we develop the assay where we can identify a nice linear range of cells that's measurable, and we study the Pressway collection of 1,200 compound as a pilot to see whether we can essentially find the chemicals that allow us to induce this population, and we use the standard deviation of 3 for cutoff. And what was interesting is just on 1,200 compound alone, we actually had a nice hit that relates to a class of compound of similar structures, which we call the inducer of cardiac progenitor expansion, that significantly increased the population by somewhere in the range of 6 to 8-fold. So we think using the mouse model that we have, we might be able to also see whether the cardiac myocyte number also expand after injury, and this just show that the false detection rate is low and significant. So for the last 2-3 slides, I want to talk about what happened then if these amount of endogenous cells are unable to repair an aging heart. So as I mentioned, the progenitor cells obviously are there in the neonatal heart, and the amounts are quite little. So a number of investigators have looked at the possibility of using embryonic stem cell as a way to regenerate damaged hearts. I won't go through all the details other than to say that these ES cells have a very nice phenotype in vitro, these round clusters, and they have very useful characteristic and ability to spontaneously differentiate into a variety of cells. And we use the GFP mouse to derive the ES cells from them, and we're able to show that if you purify the GFP cells by using flow cytometry, you get very nice beating cardiomyocytes in the dish that is quite homogeneous. And this is really the reason why people are so excited about the use of ES cells in therapy. So we have then taken on to see, is it possible you can isolate these cells and put on the engineered platform to then generate a muscle-thin film? And this was work in collaboration with Ken Chen's group primarily, and also with Kid Parker at Harvard University. So using a biomaterial where you lay down the purified progenitor cells, they eventually differentiate into nice sarcomerized cardiomyocytes. And then with that, it can be pace stimulated to go through various different cycles of systole and diastole, very similar to a neonatal mouse cardiomyocyte. And also the fourth generation of that is also similar. And we think that this would be a very nice platform for doing disease modeling or drug screen, and also for toxicity assays, since a number, probably 40 to 60%, Jay, I'm sure, knows the detail of that, of the developing drugs fall off because of cardiotoxicity, and this would be a nice assay for looking for cardiotoxic drugs. And also, eventually, we might be able to take this into tissue-based therapy. And obviously, the use of ES cells comes with a lot of controversies, and Shinya Yamanaka, who just received the 2009 Lasker Award for his discovery of the induced pluripotent stem cells, where by the introduction of 4 key reprogramming factors, he was able to take something very differentiated like the adult skin fibroblast and convert it fully into cells that behave very similar to embryonic stem cells. And amazing thing was that from his initial study 3 years ago in mouse, he and others have shown that this process could be done in human cells as well. So now there's a lot of interest in looking at the use of human iPS cells for disease modeling, taking directly from patients. And we've also looked at the characteristic for cardiomyocytes from iPS cells, which I don't have time to go over all the detail, but at least at first approximation, the iPS cells seems to look fairly similar, and they behave roughly like the way the ES cells do. But I think some of the differences may come related to the starting source. One origin, like the skin, may not necessarily be so great, whereas the origin from other cell type might be much more beneficial. And then eventually, the idea would be that we would be able to take cells from patients and go through the reprogramming process, and these iPS could then be taken, either directly differentiated into differentiated cardiomyocyte, and then go back into patient for cell-based therapy. So that is my last slide. I just want to thank the people in my lab who's done the work, particularly David Ronsley and Yuanhong Liu for the developmental work, and also the postnatal progenitors. And we have a number of collaborators who've really been instrumental in helping us with these studies. And thank you so much for your attention.